So my name is Bill Catterall. I'm a professor of pharmacology at the University of Washington in Seattle, and I'm very pleased to be participating in the iBiology program. Today, giving the third presentation in a series on electrical signaling. Our presentation today is entitled Voltage-Gated Calcium Channels, Coupling Electrical Signals to Cellular Functions. In my previous two presentations, I outlined the cellular mechanisms that generate electrical signals and allow fast control of physiological events. And we talked a lot about the voltage-gated sodium channel, which initiates electrical signals. And we've been fortunate in recent years to be able to study the sodium channel at the atomic level using the X-ray crystallography technique. In fact, the bacterial sodium channel that we used for those studies is likely the evolutionary ancestor of both sodium and calcium channels. So as you'll see, we have taken advantage of the bacterial sodium channel to learn more about calcium channels. So our focus in this talk will be about how calcium channels select calcium instead of sodium, and how they participate in cellular regulatory processes. To begin, we go back to the idea of an action potential. So in a nerve fiber, an action potential has only sodium and potassium components. But in a nerve cell body, or in a nerve terminal, or in a muscle cell, an action potential has a third ionic component. It has a calcium component. So here, illustrating the action potential in the cell body of a nerve cell, you can see that the sodium channels, again, lead the action potential and are responsible for the rising phase of the action potential. Now, the depolarization caused by sodium channels activates voltage-gated calcium channels. They let calcium into the cell. The calcium coming in also depolarizes the cell, as shown by the shaded area here. But the key thing about calcium coming into the cell is it serves as an intracellular messenger to initiate many types of physiological events. Indeed, electrical signaling would be useless without calcium channels because every physiological event that's initiated by an electrical signal is initiated by calcium coming in through calcium channels. Then, as in the nerve axon, the action potential is terminated and the cell is repolarized by the activation of voltage-gated potassium channels. From a biochemical perspective, we were able to isolate calcium channels, first from skeletal muscle, using a set of pharmaceutical compounds, the calcium antagonist drugs, as molecular bait to identify the calcium channel protein and separate it from all the other molecules in the skeletal muscle cell. And we found in those early experiments that calcium channels are multi-subunit proteins. They have a large alpha-1 subunit here, that is the pore-forming subunit of the calcium channel, and which is similar in its structure and in its function to the previously discovered pore-forming subunit of the sodium channel. So calcium channels have four other types of subunits called alpha-2, delta, beta, and gamma. They are auxiliary subunits in the sense that they're not directly involved in conducting calcium or voltage gating. But again, as for the sodium channel, these auxiliary subunits are very important for the regulation of the channel and for what you might think of as the cell biology of the channel, localizing it in the right place, putting it in the plasma membrane in the first place, and helping it to function normally. The auxiliary subunits of calcium channels are completely different from the auxiliary subunits of sodium channels, and they're very important. But our focus today is on the main pore-forming alpha-1 subunits. As I mentioned, every electrical signal generates a calcium signal by activating a voltage-gated calcium channel, and the activation of voltage-gated calcium channels initiates many crucial cellular events. And so this slide highlights secretion and synaptic transmission, contraction of muscle, regulation of many enzymes, exemplified here by protein kinases, protein phosphorylation enzymes, and even gene transcription, especially in nerve and muscle cells, very highly regulated by calcium entering the cell through calcium channels. So you can see the diversity of cell signals that are generated by calcium coming in through the same calcium channel source. So this slide kind of gives the outline of our talk. So the first part of the slide, the top part, highlights a few things that are conserved between sodium and calcium channels. And remember, they're related proteins, they do related jobs, and they have a common ancestor. So it's no surprise that much of their general mechanism is conserved. So the things that are conserved, and which I won't be talking about for calcium channels today, are the overall structure of the voltage sensing module and the pore module. These are quite highly conserved between sodium and calcium channels as far as we understand. The mechanism of voltage-dependent activation is conserved, and the mechanism of slow voltage-dependent inactivation is conserved between these closely related ion channel family members. So what I want to focus on today is what's unique about calcium channels, and especially the parts that are unique that we understand in some detail. So the first and most obvious unique property of calcium channels is calcium selectivity. They must conduct calcium ions and not sodium or potassium ions. And it's an especially hard job because outside the cell, sodium is present at 140 millimolar and calcium at only 2 millimolar. So calcium channels must select calcium in the face of a much higher concentration of sodium, and yet they must also let the calcium through really fast. So they can't just bind the calcium and let it sit there. They have to move it quickly. So how do they solve this problem? Well, to address that, we've taken advantage of their common ancestor, the bacterial sodium channel NaChBac, and we have turned NaChBac into a calcium channel by substituting the amino acid residues that are known to be important for calcium selectivity in vertebrate calcium channels. And we did that because we hoped we could create a calcium channel that would be just like a vertebrate channel, and importantly, we could crystallize it and study it at the atomic level, and that turned out to be possible. So here, in a slide entitled Converting NaChBac to CaVab, or our tongue-in-cheek name for this synthetic calcium channel, we illustrate the amino acid sequence in the ion selectivity filter and the changes we've made to change the channel from sodium selective to calcium selective. So here, for NaChBac, the key sequence is ESWSM. It also has T and L in front, but we're not going to change those. To make CaVab, we change that sequence to DDWSD. Now, if you know single amino acid code, you'll know that a D is an aspartate residue, a negatively charged residue. So essentially what we've done to make the sodium channel into a calcium channel is to change two amino acid residues from uncharged to negatively charged. We changed this key serine residue, S, to aspartate, D. And we changed this key methionine residue, M, to aspartate, D. And this changed NaChBac to CaVab, as I'll show you. We were guided in making these changes by the ion selectivity filter sequence in vertebrate calcium channels, shown for a typical human calcium channel on the right. And you can see from the underlined residues that they often have D in those key positions in human calcium channels. So did we succeed in making a calcium selective channel? So this is an electrophysiology experiment. It's carried out under what physiologists call bi-ionic conditions. So what do we mean by that? We're changing the ion composition of the extracellular and intracellular solutions to allow us to separately measure the movement of calcium and sodium. So on the extracellular side, we have 10 millimolar calcium as the only permeant ion, and we have other organic ions which are impermeant and can't go through the channel. So any ion current moving into the cell, any inward current, has got to be carried by calcium ions. Inside the cell, we've replaced all the ions with sodium, 140 millimolar sodium. So any outward going current has got to be carried by sodium. And in this way, we can judge whether a channel is carrying sodium or calcium. Now for NaChBac, the sodium channel, as you can see, the sodium currents are all heading upward in this case, and that means they are outward currents. So NaChBac is only able to conduct sodium outward in this bionic condition type experiment. It's a real sodium channel. So how about CAVAB? Well, CAVAB, just the opposite. Notice, CAVAB is only able to conduct current in the inward direction. All of these current traces are going downward to mean inward current. So CAVAB can only conduct calcium, and NaChBac can only conduct sodium. So we did achieve the change of ion selectivity. On the right side of the slide, you can see it as a function of voltage. Again, NaChBac currents only go up and out. CAVAB currents, on the other hand, come in and form a peak of calcium entering the cell. So we've successfully constructed a bona fide calcium channel in terms of its functional properties. So what structural changes did we make as we made that new construct? Well, the big thing that we did was create 2 calcium binding sites in the ion selectivity filter where there had been none before. So here on this slide, we show calcium ions illustrated by these green balls as we see them in the X-ray crystal structure of the ion selectivity filter of CAVAB. And within the ion selectivity filter, that part of the channel that interacts with and selects the ions, there are 3 key calcium binding sites. 1, 2, 3. The most important new site that we've added is site 1, which is formed by aspartate at position 178, and it used to be a serine in NaChBac. So this is the key change, and in addition, we made other changes that at this end of the channel that help to lead calcium into the selectivity filter. On the right side of the slide, we've used a technique called anomalous diffraction, which I won't explain to you, but is valuable because it directly images the calcium ion and makes sure that the image we see here in the selectivity filter is truly calcium. And you can see this selective imaging procedure for calcium also identifies sites 1, 2, and 3 in the selectivity filter. So how important are the different sites that we created? Well, the most important one is site 1. And that's a site we created by changing a serine in this position to aspartate. By doing that, we changed the channel from having a ratio of calcium permeation to sodium permeation of 0.03, so highly sodium selective, to a ratio of calcium to sodium of 30, so highly calcium selective. That's a 1,000-fold change in ion selectivity with the change of a single residue. So this is the dominant effect, creating that second calcium binding site right next to the first one, which already was present in NaChBac. Site 1 is sized appropriately to bind calcium in its hydrated form, shown here. There are 4 angstroms surrounding the calcium ion, which would allow calcium and its first shell of waters of hydration to bind effectively. We've also improved the calcium selectivity by creating a binding site for calcium at the outer end of the pore by changing this amino acid residue, which was a methionine M, uncharged, to D or to N. And this tunes the electronegativity at the entrance to the ion channel, and that's compared here in the red shading. N, which is hydrophilic but uncharged, gives light red shading at the outer end of the pore, whereas D, which is negatively charged, gives darker red shading at the outer end of the pore, indicating more electronegativity. That makes the channel 4-fold more selective for calcium, going from a ratio of 100 to 1, calcium to sodium, to almost 400 to 1. Now, 400 to 1 selectivity for calcium over sodium is just what a bona fide vertebrate sodium channel can do. So we've been able to create the bacterial channel that works just like a vertebrate channel. From that work and additional experiments that I don't have time to share with you today, we've constructed this cycle of calcium conductance. So we think then that the selectivity filter operates by alternating between having 2 ions bound in it and having 1 ion bound in it. And that's shown here. The state that has 2 calcium ions bound in it has ions bound in sites 1 and 3. We find that calcium can't bind in all 3 sites at the same time because of electric repulsion. Calcium ions have 2 positive charges, and it's energetically unfavorable for them to be near each other. So they can't occupy all 3 sites at the same time. The other important state is when site 2 is occupied, but singly. And so the channel oscillates between having 1 calcium ion in site 2 and 2 calcium ions in sites 1 and 3. And as calcium ions approach the channel, they bump one another through into the cell from the extracellular solution having a high concentration of calcium into the cytoplasm where there's a very low concentration of calcium. And in terms of a state diagram, you can see that here, the state with just 1 site occupied, the state with 2 sites occupied, and the intermediates that the calcium channel must go through to make a complete ion conductance cycle that would bring calcium rapidly and efficiently into the cell. So the calcium channel solves the problem of being highly selective by creating a calcium binding site, a second calcium binding site. Calcium binds there with high affinity and sodium does not. So sodium is excluded by high affinity binding of calcium. But high affinity wouldn't be enough by itself because the calcium would just sit there and not move into the cell. You need to have the second calcium binding site so that another calcium ion comes along and destabilizes the first one by electrostatic repulsion and pushes the first ion into the cell. And therefore, the key is to have 2 high affinity calcium binding sites closely spaced to create electrostatic repulsion and give you both high affinity to block sodium and high throughput as needed for physiology. In addition, the calcium channel makes things work even better by having 2 lower affinity sites flanking the high affinity sites to move calcium in efficiently in a stepwise process. So let's go back to our list here and look at the next topic, which is local calcium signaling complexes. So I made the point on an early slide that calcium coming in through a calcium channel does many different things, has many different effects in the cell. Secretion, contraction, synaptic transmission, enzyme regulation, gene transcription. How does the cell sort that out? If calcium's coming in everywhere and doing everything, it's not going to be a very good physiological regulator. The key solution to this problem is that all calcium signaling is local. Local calcium signaling complexes respond to local increases in calcium concentration at the mouth of calcium channels, and that calcium acts locally to do its job. So I'll give you one example of that, taken from synaptic transmission between nerve cells and either other nerve cells or their other cellular postsynaptic targets. So this slide illustrates the concept that efficient exocytosis at a synapse requires docking of synaptic vesicles within a calcium microdomain that really is of molecular dimensions. It's about 10 times the size of the calcium channel molecule itself. So it's a small space in the cell. And at an active zone, the area of a nerve terminal where calcium channels are located, synaptic vesicles are docked for exocytosis, and synaptic transmission takes place, there is a signaling complex, a very large signaling complex centered on the calcium channel as the source of calcium. And here's an example of that complex developed from proteomic experiments in which the experimenters, Müller et al., in Freiburg in Germany, detected all of the proteins they could find that were physically interacting with a presynaptic calcium channel. And amazingly, they found 100 proteins that either directly or indirectly interact with calcium channels, and some of those are shown here surrounding the calcium channel right here in the center of the action. So you get the impression then that this is really a signaling complex in which the proteins are talking to each other directly. They're not sending calcium long distances at all. So in our lab, we've been very interested in this subject as well, and we've studied members of that signaling complex that regulate calcium channel function in response to various kinds of physiological inputs. There are many examples of this, including G proteins, protein kinases, protein kinase C, the snare proteins themselves that are involved in mediating exocytosis, calcium calmodulin-dependent protein kinase, another kinase regulator, and in the example that I'll present in a little bit of detail this morning, regulation by the calcium regulatory molecule calmodulin and by related cell type specific calcium regulatory proteins called calcium sensor proteins. So all of these signaling proteins bind to sites on the intracellular surface of the vertebrate calcium channel, which is illustrated here. It's a large protein with 4 repeated domains, 6 transmembrane segments in each, and important here, very large intracellular loops inside the protein. And these intracellular sequences serve as a signaling platform for calcium signaling, binding all of these different calcium regulatory proteins that are going to carry out calcium signaling or regulate the calcium channel. So calmodulin binds to a site here in the C-terminal region of the calcium channel, and it's a site that has 2 components, an IQ-like motif called IM, and a calmodulin binding domain called CBD. Through an extensive series of studies, we've found that this interaction of calmodulin with these 2 closely spaced subsites generates a biphasic regulation of the calcium channel, which is very important in the regulation of synaptic transmission. So in this cartoon, I've illustrated the calcium channel just as the source of calcium in the surface membrane, and calmodulin here, or here, binding to its intracellular C-terminal binding site. Calmodulin has 2 lobes, an N and C terminal lobe, with different affinities for calcium. And what we found is that calmodulin bound to the calcium channel at this site can bind incoming calcium. It's sitting right there at the source of the calcium. When it binds calcium to the high affinity C-terminal domain, calcium channel activity is facilitated. So in response to local, very rapid increases in calcium, channel activity is enhanced. If more calcium comes into the cell, either because the calcium channel is open longer or it's open repeatedly, more frequently, then calcium accumulates and can bind to the second binding site on the N-terminal side of calmodulin. That allows calmodulin to interact with the second binding site on the calcium channel, the CBD, and this generates the opposite effect. This causes inactivation of the calcium current in response to longer and broader spread increases in calcium. So this flexible mechanism then allows the cell and the calcium channel to respond by increasing its activity in response to brief intense signals and by decreasing its activity in response to very long-lasting signals. Here's how this regulatory process plays out in a nerve cell in a physiological experiment. In many synapses, you see both facilitation of synaptic transmission and depression of synaptic transmission. So here, in synapses formed between two sympathetic ganglion neurons in cell culture, you can see that process. The initial synaptic response is relatively small. It reaches a peak after 3 or 4 action potentials have arrived. And then if the cell keeps firing action potentials and releasing neurotransmitter at the synapse, then the response is decreased in what is called synaptic depression. So to study this, we've been able to replace the sympathetic ganglion neuron's calcium channel, the endogenous calcium channel, with a cloned calcium channel that we can manipulate and put mutations into. That experiment is diagrammed on the top of the slide. So we inject cDNA that encodes the calcium channel, an exogenous calcium channel. We wait for 3 days. We inhibit the endogenous calcium channel with a toxin, and then we measure synaptic transmission here from the transfected channel. When we do that, with the wild type channel, we see the normal facilitation and depression that this synapse would show. If we put in a channel that is unable to respond to calcium by facilitating, we greatly decrease the size of the postsynaptic transmission and we decrease the facilitation. So facilitation of the synaptic transmission is dependent on facilitation of the calcium channel. On the other hand, if we introduce a calcium channel that has a mutation which blocks its calcium-dependent inactivation, a mutation called delta CBD here, we see plenty of facilitation, but the depression process is much increased. The size of the postsynaptic response is sustained through time much better. So synaptic depression, at least the rapid phase of synaptic depression, depends on calcium-dependent inactivation of calcium channels. This form of synaptic plasticity, as it's called, is very important in encoding information in the nervous system, and so we see that local calcium signaling and local regulation of the calcium channel is crucial for the regulation of synaptic transmission on the millisecond and second timescales. So then our last topic, again, is calcium channel regulation, but not by calcium in this case. Calcium channel regulation by signaling complexes, in this case, allows local regulation by a different regulator, by cyclic AMP and cyclic AMP-dependent protein kinase. The fight or flight response is a conserved response of vertebrates to fear, to exercise, and to stress. As you know, when you run upstairs, your heart beats more rapidly and more forcefully to push blood around and to make it possible for you to have that excess energy for exercise. So that fight or flight response at the level of the heart muscle impinges on calcium channels. The calcium channel that is in the cardiac cell and lets calcium in to initiate contraction. So here's the pathway worked out by many investigators over many years. So adrenaline or epinephrine released from the adrenal medulla, or norepinephrine released from nerve terminals, act on beta-adrenergic receptors, which activate adenylate cyclase, produce cyclic AMP, and activate cyclic AMP-dependent protein kinase. And then this cyclic AMP-dependent protein kinase acts in a signaling complex to regulate the calcium channel, which is responsible for the plateau phase of the cardiac action potential and letting the calcium in that tells the actomyosin in the muscle cell to contract. So this is a key role of the calcium channel in the heart, connecting the electrical signal of the action potential with the contraction of the heart muscle. So this process involves a signaling complex that's very well organized and very large, again, as at the synaptic, as in synaptic transmission. So the beta-adrenergic receptor, the G protein, the adenylate cyclase are all organized together with the protein kinase through binding to A kinase anchoring proteins, ACAPs, which interact directly with this C-terminal region of the calcium channel. So this calcium signaling complex is really organizing cyclic AMP-dependent regulation of the calcium channel. That signaling complex is located beyond a natural site of in vivo proteolytic processing of calcium channels, and in fact, this entire C-terminal domain is proteolytically cleaved but remains non-covalently attached to the calcium channel and crucially regulates its activity. Autoinhibition by the non-covalent binding of this distal C-terminal domain to a site in the proximal C-terminal domain strongly inhibits calcium channel function. And anchoring of all these components together by the A kinase anchoring protein, by the ACAP, is essential for this regulatory process to work in the cardiac muscle cell or indeed in the beating heart. The upregulation of calcium channel activity that mediates the fight or flight response takes place when pKa, cyclic AMP-dependent protein kinase, phosphorylates two key sites here in the proximal C-terminal domain. This part of the calcium channel is binding this distal C-terminal domain. This binding interaction mediates autoinhibition, as I mentioned on the last slide, and when those sites are phosphorylated, pKa phosphorylation relieves the autoinhibition of the channel and gives you a big increase in calcium current and a big increase in contractile force. So we've studied this process at the biochemical level with purified molecules. We've studied it at the cellular level with cells in culture. And then most recently, we've studied it in the intact mouse, where we have created a mutant mouse in which we have prevented the phosphorylation of these two key sites by a mutation of the serine residues to alanine. And so to illustrate the power of this regulatory mechanism in this last slide, we show the calcium currents measured in cardiac myocytes from a normal mouse and from our mutant mouse. So in the normal mouse, when you challenge the cardiac myocyte by beta-adrenergic stimulation, here with a drug called isoproterenol, noted on the bottom of the slide, you see a large increase in calcium current from the control to the stimulated cells. And notice again, because calcium current is an inward current, larger currents are downward. So this current is 2 to 3-fold larger than this one, and this is what mediates the increase in contractile force. Notice what happens in our mutant mice. No increase at all at this low concentration of isoproterenol, this low level of stimulation of the system. How about if we stimulate more? Well, here on this side of the slide, you can see if we stimulate more with the wild-type mice, we get an even larger response in these black symbols. And in our mice, in the mice in which the phosphorylation has been prevented, we see, again, a much smaller response, but we're beginning to see a little bit of response remaining in response in the case of this larger stimulation. So these experiments show in the context of an intact mouse myocyte and indeed in the intact mouse heart that this complex regulatory process involving a signaling complex with kinase and channel and A kinase anchoring protein and the distal C-terminus proteolytically processed and rebound to the channel, that this very complex process is indeed what is responsible for the fight or flight response in vivo. So I'll just finish up by recalling the different points from this talk. So first, and perhaps most general, calcium channels couple electrical signals to cellular functions, and their role is crucial in this regard. They're able to select calcium against sodium and other ions because they have two closely spaced high-affinity sites that bind calcium specifically, but also allow calcium conductance by a knockoff mechanism. And they have lower affinity flanking sites, which also increase the rate of calcium movement. Calcium signaling is able to be local and specific by formation of calcium signaling complexes which allow responses to locally entering calcium just around a calcium channel. And extending that theme even farther, calcium channels form signaling complexes that control not only calcium regulation, but regulation by other second messengers, and I gave you the example of cyclic AMP in the heart. So calcium channels then are the key transducers of electrical signals into physiological events and allow electrical signaling to really carry out all of the work that it needs to do in cellular signaling. Thank you very much.